Welcome back to another video, and in this video we will be learning about some of the main pointers of enzyme. In 5.1 we have the enzyme action, that says describe a catalyst as a substance that increases the rate of chemical reaction and is not changed by the reaction. That means we need to understand what is meant by the term catalyst and what is the difference in the term that says biological catalyst and the term catalyst. We are going to learn about this in the video. Next, we say describe enzyme as a protein that functions as biological catalyst and are involved in all the metabolic reactions. How enzyme is termed as protein. We have often heard enzymes said to be proteinous in nature. So we are going to learn that how it is called protein. Third one says explain enzyme actions with reference to substrate, active site, enzyme-substrate complex, and the product. We'll be learning what is a substrate, what is an active site, what is an enzyme-substrate complex, and what is meant by the term product. The fourth and the last in 5.1 says, explain the specificity of an enzyme in terms of the complementary shape and fit to an active site. That means how enzyme or the substrate is very specific in nature and how it only complements a certain enzyme to be attached to its active site. Next in 5.2 we have the effects of temperature and the pressure, how the temperature and the pressure plays a very important role as per different enzymes. Number one says, understand that the progress of enzyme catalyzed reaction can be followed by measuring the concentration of product and the concentration of reactant. Investigate. When it comes to the term investigate, it means that you have to do it with the help of some practical examination that you will be doing in the labs. And describe the effect of temperature, pH. Number 3 says, explain the effect of change in pH on enzyme activity in terms of kinetic energy, shape, and the fit. So in your syllabus, one of the terms that is very oftenly used after the being an effective collision is the kinetic energy, how the molecules gain kinetic energy and then they collide very vigorously. Denaturing and frequency of effective collision. As I just recall you about the effective collision, how the molecules frequency increase and they cause the effective collision, and how because not of the optimum temperature or the optimum pH not being provided, the enzyme gets denatured. So this is the recall in this video. What we'll be going to discuss is about the basically enzyme action and the effect of temperature and the pH. Alright. Okay. So as the first question that was being asked to us was that what is meant by the term biological catalyst. So I'll be explaining you what is the difference between the catalyst and the biological catalyst. So catalyst is something that basically speeds up a chemical reaction without being involved in it. It is not involved in any chemical reaction and it remains unchanged till the end of the reaction. How do we say that it is a biological catalyst? By biological catalyst, it means that it is present in all the living cells. And by living cells, I mean that it is present in certain plants, it is present in certain animals, let's say kangaroos, let's say a bat, let's say a cat, or any animal. Then let's say it is in living cells, we include bacteria. In living cells, we also include fungi that may be unicellular or it may be multicellular. Let's say yeast, which is a unicellular, or let's say mushroom, which is basically a multicellular. Or we can also talk about Protoctis. How do we know that Protoctis is also a living organism as it is unicellular in nature? For example, let's take an example of amoeba. This is the basic definition or the basics difference between a catalyst and the biological catalyst. That the biological catalyst is mainly present in all the living cells, or all the plants, or all the animals, bacteria, fungi, or the prototypes, whether they are unicellular or whether they are multicellular. Whereas when you talk about catalyst can be anything that can speed up a chemical reaction without being involved in it. It is not majorly required that its composition should be included in living cell. It must be included in non-living cell or any element or any compound. So that's it. That we will be talking more about the biological catalyst in it. That how the biological catalyst that we say an enzyme basically work very best. So an enzyme is proteinous in nature. When we say that it is proteinous in nature, what is meant by that? That it is made of protein. And the subunit of protein is amino acids. Thus, the enzyme in turn is made up of amino acid that joins together to form a protein. Thus, we say that enzymes are proteinous in nature. Next, a very important point of enzyme is its specificity. How enzyme is very famous for its specificity is because not all the enzyme or not all the substrate can be attached to the active side of the enzyme. Let's discuss about what is meant by an active side. I've discussed this in the previous video, but for the review, we will be discussing about an active side. Active side is basically a depression or a groove on the outer surface of enzyme where the substrate is allowed to be attached and form as enzyme-substrate complex. Now let's discuss what is basically meant by enzyme-substrate complex. So, enzyme-substrate complex is a term. When we talk about it, it means when an enzyme is combined with a substrate, it forms an enzyme-substrate complex, which is a phenomena of binding of an enzyme with the substrate, in turn finding a product finally. So, our need is to complete a reaction and basically form a product. But before the formation of the product, the step in which the enzyme is being attached to the substrate and the reaction is being held, it is known as the enzyme-substrate complex. This is a substrate, this is an enzyme. It has an active site where the substrate is going to be attached and the reaction shall take place in order to form a product. Another point of an enzyme that is very often asked in the paper is what is meant by the optimum temperature or pH. Let's discuss about the term optimum. Now, what is meant by the term optimum? The term optimum means anything that works best at a certain temperature or a certain pressure or a certain pH. When you talk about the human body optimum temperature, for example, we say is 37 degrees centigrade. That means its regulation or the best amount of the work is done on the temperature when it is provided at 37 degrees centigrade. Do remember that each enzyme varies within optimum temperature or pH. It is not important that if an pepsin works best at 1.5, the amylase would also work best at 1.5. It might work best on 3.5. This is very often asked in the question that you are given some number of enzymes and some number of pH. We do have to learn that the very basic pHs of certain enzymes. Let's say that I have given an example of pepsin. For say pepsin works best at 1.5. Let's say salivary amylase works best on 3.5. So that's it about an optimum, the term what is meant by an optimum temperature. Let's discuss about what is meant by the optimum temperature, particularly in detail. So we can be learning about it by the help of a graph that says that this is a graph, this is its highest point, and by the highest point it means it is working best at this point. So its optimum temperature at this point, let's say, is 30 degrees. The graph over here shows that it progresses this way, the energy has been providing this way, and the reaction forms up a product once the reaction is completed. At this point, we will be learning of how do we know that the optimum temperature is 30 is due to when it has reached its optimum point, it starts to decline. What does it mean when the enzyme starts to decline? It means that enzyme starts to become denatured. Now, the term denatured means when the temperature raises up to 30 degrees centigrade, the enzyme is not able to work at its best. Thus, it becomes denatured. Or when the enzyme decreases in its optimum temperature, then also it is not able to work very well, and we say that it becomes inactive. So from all of this, we learned that there is always a certain amount of temperature that is required by an enzyme in order to work its best. To reach the temperature and then to be declined, we need that temperature at which it works very efficiently. Same goes with an optimum pH. When you talk about optimum pH, it is a certain amount of pH, let's say 7. 7 works best when it comes to water as it is a neutral quantity. But for certain enzyme, let's say pepsin works best at an acidic temperature, acidic pH. So it requires acidic pH in order to work its best. If it is not provided with the acidic pH and it is provided with the alkaline pH, then it does not work very well. One thing that I need to clear out over here that is related to the term denatured. Sometimes the students do confuse the word denatured as the enzyme is getting killed. So this is not what a denaturing means. Denaturing means that the shape of an active site is basically lost. Shape of an active site is lost. It does not mean that the enzyme is being killed. It means that enzyme do work, but it works very inefficiently. And it does not work very best. This is a very common question that is being asked, a very common confusion that a student usually face. Another very common mistake that we often make is that this is an enzyme, this is also an enzyme, this is also an enzyme, this with this and also an enzyme. What do we need to learn by making such disastrous diagrams? What do we need to learn is that each enzyme or different enzyme has different specific shape, which also determines it to be very particular when it comes to a substrate. Not, for example, this is a substrate, all right? This substrate cannot be attached to this. This substrate cannot be attached to this. This substrate neither can be attached to this, but it can be attached to this. Thus, this substrate is meant to be attached to this and neither of the other enzymes. We need to learn that each enzyme has a specific shape of its active site. Thus, it requires a certain substrate to be attached to it. As we have learned earlier, when the enzyme combines with the substrate, it forms an enzyme-substrate complex. Why I'm repeating it again is that this is a very important term when it comes to Olevels, as these terminologies are being used over here in your syllabus. The last thing that we shall discuss in the revision video is the concept of lock and key. We have discussed this very earlier and we'll be revising is what is meant by the lock and key hypothesis when it comes to the chapter enzyme. So basically, I'll be giving you an example and we'll be learning with help for that. Let's say starch is converted into maltose with the help of enzyme, let's say amylase. All right, and let's take another example. We say that the maltose is being converted into glucose with the help of an enzyme that says maltase. What we need to determine is that what is the lock and what is the key in these reactions. Always that we need to remember is that lock is an enzyme, whereas key is always the substrate. If you students remember this trick, it will be very easy for you to solve all of such MCQs. When we say lock is an enzyme, that means all of the enzyme we already know that an enzyme has a A's in it. If you don't remember that always on the arrow is an enzyme written, we do know, and we do also remember that the term A's is always added to a enzyme amylase maltase. So this is an enzyme, that means this is a lock. The next thing that we need to determine is about the substrate and about the product. We already know that at the end of a reaction, we find a product. This is easier to determine when it comes to the reaction. We say that this is a product and the rest that is left is obviously the substrate. We say that the starch is a substrate. Let's recall it. When a starch is converted into maltose in the presence of an enzyme, which is the amylase. So amylase is an enzyme, as we know that an enzyme is a lock. Thus, the amylase is a lock and a starch is a key. Since the substrate and the enzyme form a enzyme-substrate complex, and that in turn forms a product, thus the maltose is a product. Now you can solve this equation very easily. That says maltose is a substrate, maltase is a lock, and the glucose is the product. All of these are very important for us to understand. Let's come back to this page and recall it that we have discussed all of these points when it comes to 5.1 and the 5.2. So the enzyme action, in which we have discussed that what is a biological catalyst, what is the catalyst, as we have discussed earlier. We have also discussed why the enzymes are proteinous in nature. We've also discussed what is a substrate, what is an active site, what is an enzyme-substrate complex. Let's recall it. When the substrate is attached to an enzyme, it forms an enzyme-substrate complex, which in turn forms a product. And the groove and the depression, or a certain amount of place that is present on the enzyme, we call it as an active site where the substrate is being attached. Lastly, it says to be discussed is the specificity of enzyme, and we have discussed how there are different shape of enzyme and only certain or a specific substrate can be attached to an enzyme. In 5.2, we have discussed about the effect of temperature and the pH. Also, we have also discussed about the optimum temperature and the optimum pH of an enzyme. So, we've also discussed about the concentration of product and the reactant. We have discussed about the effect of temperature and the pressure. We've also discussed about the denaturing and how the molecule's kinetic energy increases, thus the frequency of the effective collision decreases. All of the points in 5.2 and 5.2 has been covered. I hope you find this video very helpful.